As a part of the retina, the bipolar cell exists between photoreceptors and ganglion cells. They act, directly or indirectly, to transmit signals from the photoreceptors to the ganglion cells. Overview. Bipolar cells are so named as they have a central body from which two sets of processes arise. They can synapse with either rods or cones, and they also accept synapses from horizontal cells. The bipolar cells then transmit the signals from the photoreceptors of the horizontal cells, and pass it on to the ganglion cells directly or indirectly. Unlike most neurons, bipolar cells communicate via graded potentials rather than action potentials. Specification. Bipolar cells receive synaptic input from either rods or cones, but not both, and they are designated rod bipolar or cone bipolar cells respectively. There are roughly 10 distinct forms of cone bipolar cells. However, only one rod bipolar cell, due to the rod receptor arriving later in the evolutionary history than the cone receptor. In the dark, a photoreceptor cell will release glutamate, which inhibits the on-bipolar cells and excites the off-bipolar cells. In light, however, light strikes the photoreceptor, which causes the photoreceptor to be inhibited due to the activation of opsins, which activate 11-trans-retinal, giving energy to stimulate G-protein-coupled receptors to activate phosphodiesterase, which cleaves cGMP into GMP. In photoreceptor cells, there is an abundance of cGMP in dark conditions, keeping cGMP-gated dark channels open, and so activating PDE diminishes the supply of cGMP, reducing the number of open dark channels and thus hyperpolarizing the photoreceptor cell, causing less glutamate to be released. This causes the on-bipolar cell to lose its inhibition and become active, while the off-bipolar cell loses its excitation and becomes silent. Rod bipolar cells do not synapse directly onto ganglion cells. Instead, rod bipolar cells synapse onto a retina amacrine cell, which in turn excite cone on-bipolar cells and inhibit cone off-bipolar cells, thereby overtaking the cone pathway in order to send signals to ganglion cells at scotopic ambient light conditions. Off-bipolar cells synapse in the outer layer of the inner plexiform layer of the retina, and on-bipolar cells terminate in the inner layer of the inner plexiform layer. Functionality. Bipolar cells effectively transfer information from rods and cones to ganglion cells. The horizontal cells and the amacrine cells complicate matters somewhat. The horizontal cells introduce lateral inhibition and give rise to the center surround inhibition, which is apparent in retinal receptive fields. The amacrine cells also introduce lateral inhibition, however, its role is not yet well understood. The mechanism for producing the center of a bipolar cell's receptive field is well known. Direct innervation of the photoreceptor cell above it, either through a metabotropic or ionotropic receptor. However, the mechanism for producing the monochromatic surround of the same receptive field is under investigation. While it is known that an important cell in the process is the horizontal cell, the exact sequence of receptors and molecules is unknown.